What if I told you that scientists can now edit the DNA of a living human being with the precision of a word processor? That we can literally copy, cut, and paste genetic code like editing a document. This isn't science fiction. This is CRISPR gene editing, and it's happening right now in laboratories across the world. Picture a molecular pair of scissors that can find any specific sequence in your 3 billion DNA letters and make exact cuts. That's CRISPR. And it's not just cutting, it's rewriting the fundamental code of life itself. This matters because we're standing at the threshold of eliminating genetic diseases that have plagued humanity for millennia. Huntington's disease, sickle cell anemia, cystic fibrosis. Conditions that destroy lives and families could become medical footnotes. But we're also facing the possibility of creating genetically enhanced humans. Designer babies with superior intelligence, perfect health, and extended lifespans. The clock is ticking. China has already created the world's first gene-edited babies. Other nations are racing to catch up. The question isn't whether this technology will reshape humanity. The question is whether we'll control it, or whether it will control us. So how does CRISPR actually work? Imagine your DNA as a massive library containing 3 billion books. Each book represents a gene. Now imagine you need to find one specific sentence in one specific book and replace it with a better sentence. In a traditional library, this would take decades. With CRISPR, it takes hours. CRISPR stands for clustered regularly interspaced short palindromic repeats. Think of it as a molecular GPS system combined with precision scissors. The system has two main components. First, a guide RNA that acts like a bloodhound, sniffing through your DNA until it finds the exact genetic sequence you want to change. Second, an enzyme called Cas9 that functions as molecular scissors, cutting the DNA at precisely the right spot. Picture a factory assembly line, but instead of cars, it's producing proteins. Your genes are the instruction manuals for this factory. If there's a typo in the manual, the factory produces defective proteins, leading to genetic diseases. CRISPR finds these typos and corrects them in real time. The process begins in a laboratory. Scientists design a guide RNA that matches the problematic genetic sequence. This guide RNA is like a key that only fits one specific lock in your entire genome. They inject this guide RNA along with the Cas9 enzyme into cells. The guide RNA searches through millions of genetic letters until it finds its perfect match. When it locks on, Cas9 springs into action, cutting both strands of the DNA double helix. Now comes the magical part. Cells have a natural repair system. When they detect broken DNA, they immediately begin fixing it. Scientists can hijack this repair process by providing a template with the correct genetic sequence. The cell uses this template to repair the cut, effectively replacing the problematic gene with a healthy version. Imagine editing a Wikipedia article, but instead of changing text, you're changing the instructions that determine eye color, disease resistance, or cognitive ability. The edit becomes permanent, passed down to every cell that divides from the original. But here's where it gets controversial. In 2018, a Chinese scientist named He Jiankui shocked the world by announcing he had created the first gene-edited babies. Twin girls named Lulu and Nana were born with modified genes that theoretically made them resistant to HIV infection. The global scientific community erupted in outrage. He Jiankui was imprisoned and his career destroyed. But the precedent was set. The first genetically modified humans were already walking the earth. This raises profound ethical questions that humanity has never faced before. Should parents have the right to genetically enhance their children? If genetic enhancement becomes available, does that create a two-tiered society of the genetically enhanced and the natural born? What happens to human diversity when parents can choose their children's traits from a genetic menu? Consider this scenario. Wealthy families start genetically enhancing their children for intelligence, athletic ability, and disease resistance. These enhanced children grow up with significant advantages over natural-born children. They dominate universities, athletics, and eventually the job market. Within a few generations, genetic enhancement becomes mandatory for competitive success. We could be creating a world where your genetics determine your social class more rigidly than ever before. The enhanced elite versus the genetic underclass. This isn't speculation. This is a probable future unless we establish global regulations now. But the potential benefits are staggering. CRISPR could eliminate genetic blindness, allowing millions to see for the first time. It could cure sickle cell disease, which affects millions primarily in Africa and among African Americans. Scientists are already using CRISPR to develop new cancer treatments by editing immune cells to better recognize and destroy tumors. Imagine a world where genetic diseases are extinct. Where cancer becomes as rare as polio. Where aging slows dramatically because we've edited the genes responsible for cellular deterioration. This technology could extend human lifespan to 200 years or more. Agricultural applications are equally revolutionary. CRISPR-edited crops could survive extreme weather, resist pesticides naturally, and produce higher nutritional content. We could feed the world's growing population while using less land and water. Looking into the future, CRISPR will likely become as common as vaccination. Genetic counselors will work with expectant parents to identify and correct genetic problems before birth. Gene therapy clinics will treat diseases by editing patients' DNA directly. The technology is rapidly becoming cheaper and more accessible. What once required million-dollar laboratories can now be done with equipment costing thousands. Biohackers are already experimenting with DIY genetic modifications in garage laboratories. Within 20 years, we'll likely see the first generation of intentionally enhanced humans reaching adulthood. These individuals might have perfect vision, superior memory, enhanced physical strength, and resistance to major diseases. They'll represent humanity's next evolutionary step, but it will be guided by human choice rather than natural selection. The question facing humanity isn't whether we should develop this technology. It's already developed. The question is how we'll use it responsibly. Will we create a better future for all humanity, or will we fracture our species into genetic castes? Every day we delay establishing international ethical frameworks, we move closer to an unregulated genetic future where the highest bidder determines the course of human evolution. The decisions we make in the next decade about CRISPR will echo through centuries. We're not just editing genes. We're editing the future of our species. And that future is being written right now, one DNA letter at a time. Subscribe to VoidViz to decode tomorrow's tech.